Hi, I'm Russ of Aquarimax, and in today's installment of the Isopod Care Guide, we'll talk about breeding Isopod Morphs. Simply breeding isopods, by which I mean just getting them to produce offspring, is straightforward with many species. Generally, you just keep several males and females together under good conditions, and they'll breed. However, breeding them selectively is a little more complicated. One reason for this is that both males and females of many species are reproductively mature at about a third of their adult size, and so they'll start to breed at pretty young ages unless you separate them. Another complication is that females can store sperm for multiple broods of young after mating just once. You can't just pair an adult male and an adult female that have been kept with other adults and expect the female to produce your chosen male's offspring. Then there's the fact that some morphs within what are currently considered species are not compatible, like Porcelliolavis dairy cow and Porcelliolavis orange, at least the forms that are prevalent in the US. They'll try to mate, but there's something that prevents them from producing fertile offspring. There are different ideas on why this might be the case, but that's for another video. It's quite possible to distinguish male isopods from female isopods. Some species have very obvious distinguishing characteristics in mature males, like the extended exopodites on the uropods of the males of some Porcellio species. Others have more subtle distinctions. There are various characteristics that can help, but the most dependable way to determine whether an isopod is male or female is to look underneath the pleon, or the rear portion, if you will, at the pleopods. Here you can see that males have modified pleopods used in mating, and a female does not have this modification to the pleopods. In all honesty, when I want to breed isopods selectively, I rarely concern myself with distinguishing male from female and separating them into pairs. This is because there are easier ways to go about selectively breeding isopods. In fact, the best way to explain it, I think, will be to describe step by step how I combine two single gene recessive traits of Porcellioscaper. This basic method can be used for combining any single gene recessive traits and modified somewhat for other selective breeding projects. A few years ago, when the isopod hobby was significantly smaller than it is now, a hobbyist by the name of Ryan Orr produced the first orange Dalmatians. I was following along with his progress of this project, so I decided to try to produce some myself as well, and this is how I did it. First, I selected a number, and I think it was about 16 each, of very small individuals of both P. scaber orange and P. scaber Dalmatian. Choosing very small, immature individuals ensures that they have not yet mated with individuals of their own morph. And good numbers of each of them greatly increases the chances that some of them will cross with the other morph. After housing the two morphs together, I simply waited for them to mature and start producing offspring. Well, let me take a step back here. I think understanding of how the orange and Dalmatian genes work would be useful. So my understanding is that the gray pigment in many isopods is the result of a modification to an already existing orange pigment. In other words, orange is a step on the way to gray, so that any mutation that interferes with the transformation of that orange to gray will result in orange specimens, and this helps explain why there are so many isopod species with orange morphs. Dalmatian, on the other hand, as I understand it, interferes with the distribution of the gray pigment. So Dalmatians produce the gray pigment, but it doesn't get transported very effectively, which is why the random gray spots can be very large or very small, and some Dalmatians have very little gray at all. Again, all it takes to produce a Dalmatian, or a pied morph, is something that interferes with the distribution of pigment, and there are quite a few pied isopods out there. Before I get back to my orange Dalmatian project, I want to thank my patrons at Patreon. Your contributions to this channel are incredibly helpful, and I am very grateful. For as little as $1 a month, you can help too. If you're interested, please click the link at the end of this video. So now, back to my orange Dalmatian project. As I said, I had put some very young orange and Dalmatians together, and I was waiting until I saw offspring. Dalmatian or orange offspring that showed up in this enclosure were obviously not crosses between the two morphs. What I was keeping my eye out for were visual wild types. Since both Dalmatian and orange are recessive traits, one would predict that a first-generation cross between the two to be 100% wild types in appearance. Interestingly, I did not see exactly those predicted results. The vast majority of the offspring were indeed gray wild types, and I certainly didn't see any Dalmatians or any oranges. I did see a few calicos though, which was a little surprising, but that's a tangent and I'll talk a little bit more about calicos later. So as soon as it was practical, I separated the young wild types from the parents. I didn't want the parents to mate with the offspring because such a pairing would delay my results. It is important to note though that these gray specimens were visually wild types. They were heterozygous for both the orange trait and the Dalmatian trait. So they, in other words, carried a copy of each of those genes. Specimens that carry two recessive traits like this without expressing them visually are known for short as double hets, a term which selective breeders of reptiles and various other creatures will be familiar with. So once these double het offspring matured, they began to reproduce, and many of their offspring were visual wild types carrying the orange or the Dalmatian trait. And some of the offspring were orange, others were Dalmatian, and about 1 in 16, statistically speaking, were orange Dalmatians. So I began to separate the orange Dalmatians as soon as they were distinguishable from the other offspring. At this point, I had succeeded in producing my own line of orange Dalmatians. I later obtained some of Ryan Orr's stock and combined my stock with his, so the orange Dalmatian that I now keep and sell are descended from both lines. As I mentioned earlier, working with any two single-gene recessive traits with genetically compatible isopods is done in essentially the same way. For instance, so far it appears that all of the color morphs of Porcellionides pruinosus are genetically compatible. Kyle of Roach Crossing has crossed the pied morph, known as Oreo crumbles or cookies and cream, and the orange morph to produce a morph known as orange cream, and I have heard that others have done the same. In fact, I have begun this process myself as well. I put some very young individuals of Oreo crumbles and powder orange together, and they're now beginning to produce offspring. I should soon begin to be able to separate out the double het offspring, which is half the battle. Many of the isopod traits in the hobby, like the ones we have discussed so far, are single-gene recessive. However, there are some that are not. For example, there are several lines of calico Porcellioscaper. Some are regarded to be sex-linked in that the males do not express calico patterns, and the females do, while others do not appear to be sex-linked. The particular strain of Calicoscaper I keep produces oranges, wild types, and calicos of various colors and markings. On the other hand, some strains, like orange calico, produce few, if any, wild types. In any case, calico seems to be more genetically complex than a single-gene recessive trait. Porcellioscaper Lava is a relatively new morph that I obtained from SmugBug. It's indicated on the website that this morph has been shown to be codominant. If lava works the same way as other codominant genes, a specimen with the lava trait crossed to a pure wild type will produce offspring in the first generation with some lava markings, although in the next generation, if those offspring are crossed, individuals with two copies of the lava gene will be superforms, meaning that the expression of the trait will tend to be more pronounced. My lava colony is still new, but I have a lot of tiny offspring that have appeared, and I'm excited to work more with this trait. There are also traits that appear in isopods from time to time that they do not pass on to their offspring, or at least not very dependably. Just to cite two examples, there was a pied form of Silisticus convexus some years ago that Invertebrate Dude was working with. And there was someone, I'm not sure who, that was working with a specimen of Porcellio dilatatus that was white. And neither of these became established in the hobby because their descendants, even after multiple generations, did not maintain the trait. Fortunately for us, another pied form of Silisticus convexus appeared later, which does breed true, and it's now known as Ukraine pied, and I now have specimens of that morph from SmugBug. There are also line-bred traits. These are traits that can be selected for over generations, but it's clear that they are not as easily passed on as single-gene traits. It may take many generations to produce individuals that consistently show the genes, or consistently show the traits that you want. An example of this is the Armadillidium vulgare night gold line that I've been working on for years. By carefully selecting individuals with the coloration I'm looking for, I've increased the frequency and intensity of the coloration in the population, but the trait is far from consistent, and may never be. If you happen to encounter an unusual specimen among your isopods, and you want to see if you can isolate that trait, you can separate that individual with a small number of other specimens, maybe 6 to 10, from that same culture. And this can help increase the chances that the trait will show up again in a generation or two. And once you have a number of specimens with the unusual trait, you can start separating the individuals with that trait from the others. That's what I'm currently working on with specimens like these, which began showing up in my Armadillidium vulgare orange vigor culture. Genetics are quite fascinating, and this has just been a very brief introduction to the basics of working with genetic traits in isopods. I'd love to hear about your isopod breeding projects. Let me know what you're working on in the comments. Thank you for watching. I post videos every Tuesday and Friday all about aquarium and vivarium pets, with lots of isopod content. Feel free to share, rate, comment, and if you haven't already,
subscribe.
And then tap the bell so you don't miss my next video.